My name is Sean Gallagher, and I am with the American Association for the Advancement of Science, or AAAS. We are the largest general scientific society both in the country and the world, and our mission is dedicated to the advancement of scientific knowledge for the good of society as a whole. And today's briefing is a perfect example of the type of work that we do, and we're very happy and glad that you have decided to join us on this very important topic. This is the 6th year of the series and the 3rd briefing of this year. And so we hope that you'll stay tuned for more to come. We usually do about 4 or 5 of these per year. And we love feeding you, so eat up. A big thanks to the Congressional Neuroscience Caucus, who we work with, and the staff of Kathy McMurray-Rogers and Earl Blumenauer, who help reserve these rooms and get the word out about our speakers. I am personally delighted to have such an excellent lineup of folks today to talk to, these 2 speakers. You should have a list of them with you with all of the information, and I'll let them do the introduction because they're going to do a lot better job than I am describing what they do than I would. So again, please continue to eat, and we'll be following up with all of you with information on the speakers. This is being recorded as well, so if you have to leave early, or if there's anybody else you know that would be interested in watching this, we'll have a live video stream available next week. So without further delay, I'm going to call our first speaker up, Dr. John Brownstein, who is the Chief Innovation Officer at Boston Children's Hospital and Professor at Harvard Medical School. Dr. Brownstein?
Great. Thank you, and thanks to AAAS for inviting me here today to tell you a bit about some of our work in public health surveillance, and specifically around Zika. I thought it would be helpful to start the talk to give you a sense of the issues around public health surveillance and actually understanding emergence of viruses that take place in populations, and why there are so many challenges around understanding what's actually happening with Zika as it emerges across Central and South America and likely into the US. So when we think about the weather, we have apps on our phone. Obviously, it's not 48 degrees right now in Boston. But the idea is that there is a huge amount of data that is collected from agencies like NOAA that are then pushed out into APIs, which are a way of transferring that information that allow us really to understand what's happening in populations and build forecasts of potential weather conditions. So the real issue is when it comes to public health, we're missing a lot of these components, right? So we're missing the actual underlying data. Now, agencies like the CDC and others attempt to do surveillance, but globally, this is, of course, a real challenge. There's no sort of ability to extract that data, even if it did exist, and then there's sort of no ability to then build those forecasts and have a tool that enables governments, but also the public, to understand what's going on the ground. And so a lot of our work over the last decade has really been geared towards understanding new data streams, new sources of information that can provide us really important insights into public health issues that are taking place around the world. So I think you'll probably recognize some of these logos. The idea is looking for patient-generated data that's coming from either the population with crowdsourcing tools or through experts to allow us to get insights, especially into places with limited public health infrastructure. So our goal, and I'll tell you a little bit about how we do this and then how we apply this to Zika, is to take the massive amount of information that's coming via the web, via social networks, and try to organize that in a way that allows us to understand issues that are taking place in populations. So the idea is, of course, if people are tweeting or talking about issues on the ground, about, for instance, symptoms that might relate to fever or unknown outbreaks that are taking place in populations, we might be able to pick up these issues sooner with these kind of data. And even beyond that, the idea is that these are data that don't actually necessarily have borders. So we can actually collect and get insights into public health issues in places with limited infrastructure or potentially limited transparency around what's happening on the ground. So this is an area, a field called, we've termed digital disease detection. It's been emerging over a decade or so. And the idea is that it can transform the way that we get insights into public health issues. And so the work that we've been involved in, and this is among an ecosystem of tools that are out there, is a tool called HealthMap. And basically, it's essentially trying to be the weather.com for disease outbreaks. The idea is that we're mining and constantly scraping the web, and essentially like Google for disease outbreaks, mining through many sources of information, hundreds of thousands of websites across many languages to get insights into disease outbreaks. And so the idea is that the first signs of an event may come from either an astute clinician, a reporter, someone on the ground. And the idea is that through tools like natural language processing and machine learning, we can actually begin to take that huge amount of data that's being sort of appearing online, we call digital exhaust, attempting to bring that to bear. So natural language processing means the idea of taking free text, whether it's a tweet or a news article, and taking that information and turning it into a database where we have information about diseases, case counts, locations, and then using tools of essentially artificial intelligence or machine learning, we call, the idea is that we can actually start to categorize this information and look for particular signs of emerging issues in populations while removing a lot of the noise that comes from these tools. And so that's really what the focus of our efforts have been, and really what's provided insights into organizations like the WHO, like CDC, who are using this information on a daily basis to get insights into what's happening in the population. And this is sort of HealthMap tool, and the idea is attempting to be that information source to communities to understand what's happening locally, so what are the outbreaks that are happening around me, but also give insights to what's happening around the world. So before I get to Zika, I'll just mention there's been many examples of how this has worked. So swine flu, if you remember, some of the first signs of what was taking place in Mexico, in Veracruz, didn't come through official channels like the Mexican government talking about the earliest signs of H1N1, but actually came up from a local reporter talking about a mysterious illness in a particular town. And from this kind of data, we can actually track a virus in real time. So we can use the collection of all these sources to basically track how a virus is moving from location to location. If we fast forward from swine flu into more recent times, and for instance, the emergence of a new flu virus in China, H7N9, we can start to mine sites like Twitter, but also Chinese sites like Weibo, and identify cases that are emerging well before government officials are talking about them. And then, of course, even more recently with the emergence of Ebola across West Africa, we recognize that the mining of information from people on the ground, from NGOs, from a variety of different sites, we can actually produce a real-time view of what's happening that is giving government officials situational awareness, not only about the region, but when you connect that information to transportation patterns and climate and weather, you can begin to build forecasts of what's happening and what could happen down the road. Okay, so now we turn to Zika, and essentially Zika it represents an important, of course, emerging disease transmitted by mosquito vectors. So Aedes aegypti is the mosquito vector, actually discovered many decades ago in Uganda and isolated in Nigeria in 1954. We started seeing outbreaks emerge in 2007, and then, of course, more recently, the emergence across Central and South America in 2014, likely tied to the World Cup. And of course, we know that there's been an incredible amount of movement of this virus because of the prevalence of this mosquito vector. And so the way it works is a mosquito carries the virus, transmits it to a human, and then other mosquitoes would bite that human and get infected, and that's how you get chains of transmission that take place. And so this is an emerging virus, but we see many other emerging mosquito-borne viruses, flaviviruses they're called. West Nile virus, you might remember. Dengue is very similar, and so chikungunya is another one that you may have heard of. So this is something that, of course, has happened over and over again. Of course, the concerns around pregnancies and infants is really what elevates Zika beyond some of these other types of events. And so our perspective, and the next talk will be much more geared to the therapeutic side, is really about the issues around surveillance. And because we need situational awareness to understand how to mobilize public health efforts, you need good surveillance. You need to understand what's actually happening on the ground in order to basically direct, for instance, mosquito control efforts and eventually potentially vaccine efforts. And we know that public health just generally, globally, not just in the US, is sort of dwindling in terms of skilled workforce, in terms of methodologies to collect data, and then the response side, which often is just about mosquito control. Many of these programs have been eliminated or just not at the level that they need to be to control these outbreaks. And so many of these countries are really struggling to actually control these events. So our effort has been just about mapping and how do you just build a view of what's happening on the ground. And so our team in Boston, at Harvard and Children's Hospital Boston, has really been focused on collecting data from all these sources to build essentially a living, breathing map of what's taking place around the world. And so we have a team of experts, public health students especially, that are curating massive amounts of data daily to try to provide sort of the best situational awareness view of what's taking place. In addition to that, there's a huge amount of effort to actually understand the Aedes mosquito and where it's distributed, specifically Aedes aegypti, because if we know where the Aedes mosquito lives, then we have a better understanding of where future risk might be. Of course, we're not going to see cases in place that's missing this important vector, although there have been reports of other types of mosquito species, in particular Aedes albopictus, being potentially a vector as well. But for now, the idea is to focus on places where that mosquito lives, although that's a changing range of habitat based on, of course, changing climate, and basically figure out how to begin to expect where risk might emerge in populations. So our efforts have been all about trying to predict and forecast where there might be future risk by analyzing as much data as we can access. And beyond that, from the data that we have about cases, but also ranges of Aedes aegypti, climate, landscape, all these variables come together for us to build models of where we might expect future risk. So you can see many of the places we know that there's existing Zika virus transmission are highlighted on this map. There are many places that have yet to experience chains of transmission. You'll see the southern part of the US. That does represent, of course, a risk for transmission. Now, there's still, it's still unclear how much risk we'll see in those parts of the US, especially based on the fact that the behavior of populations here is slightly different than populations in Central and South America, especially with prevalent AC and just generally air conditioning. And so essentially we still don't know fully how bad it will be. We will likely see cases, but probably not to the same extent as other parts. This will follow the similar trajectory of things like dengue, chikungunya, which have not seen major outbreaks. We've seen cases and some level of transmission, but not the same level as potentially other types of viruses. And so from there, we've been building out our interface and working with government officials to essentially have this better situational awareness. The other aspect of the work that we're involved in is building risk assessment based on transportation networks. So you can imagine a huge part of what we're interested in is figuring out where the virus might move based on where populations are moving. And so we've been building predictive models based on very detailed passenger itinerary data from places from Brazil to other parts, for instance, in the US, to think about how many people are moving to those places, what is the likelihood that, and the probability of someone from one of those regions that will be carrying the virus, or potentially even a mosquito hopping on a flight and getting to a particular location. And a lot of our focus, of course, is going to be around the Olympics. We spend a lot of time doing mass gathering surveillance. So that's the idea that whenever there's a mass gathering, you have this risk of huge populations moving into a particular location and bringing in potential pathogens, and of course then the export of those pathogens to around the world. So what we end up doing is spending a lot of time focusing our surveillance not only just at the site of that event, but also the places that are most connected to those events. So we get data on transportation networks, of course, but also information about visas that are issued or likely attendees from athletes. And from there, we can start to make predictions of where we might see movements. We've done this for things like MERS and other types of viruses, and we'll do the same again for the Olympics that are coming up. I want to just mention a little bit in terms of sort of innovations in public health, because a lot of our challenges are just in the ways in which data is collected. It's a real challenge to get data from patients that then moves to hospitals, that moves to local public health, and then eventually gets filtered up to the CDC. That can take a huge amount of time. And so a lot of our work is focused on why don't we just ask the public directly to get the public involved in surveillance and really much more engagement out of individuals, because of course there's so many concerned citizens. Our goal is sort of, we say, putting the public back in public health and really make it much more about engagement rather than just making individuals a source of data for public health surveillance. So we stood up a system called Flu Near You, which was initially geared towards, of course, influenza. We have over 100,000 people registered in the system, and essentially they provide us information about symptoms that they're experiencing on a weekly basis. And from there, we can get important insights into, for instance, influenza outbreaks as they emerge in populations, just simply by asking people to spend a few seconds every week to tell us how they're feeling or how their family members were feeling. But from there, we get very important demographic and geographic data, as well as vaccination status, so we can actually quickly calculate the vaccine efficacy on a given season based on what people are reporting. And the cost of this system is a fraction compared to maybe more other types of CDC-based surveillance systems that take a huge amount of infrastructure to collect that data. What we're doing now, and we've actually implemented this, is we've expanded our system, Flu Near You, to then actually include symptoms that might relate to Zika virus. And so we've now started this as an effort to basically, as we expect Zika to emerge in the US, using our sort of individuals that are part of the system as sentinels for what's happening on the ground, and especially in areas of the southern US where we might expect cases to emerge. So that's TBD, but the idea is that we're going to likely have a tool that will enable us to have quick insights into what's happening on the ground. And then we have an internal dashboard that we provide to public health officials that allow us to very quickly see much more granular data about what people are reporting, in addition, of course, to our public website. We're spending a lot of time at Boston Children's thinking about new tools and new ways of engaging the public. And so, for instance, this is a fever calculator that we developed. It's called Thermia, and it's the idea that people can find out what a fever means and get sort of recommendations from Boston Children's about what to do in the case of a fever for yourself or for your child. But what this does is actually provides us underlying data about what's happening in population. So we can see spikes of potential febrile illness that might, of course, relate to Zika or other types of infections. And provide us with underlying data as well, but by giving people important information about how to treat fevers. And we're actually connecting this data to devices. So I think what we're seeing now is an explosion, of course, in the passive collection of data. These are a few different devices that are out there on the market right now that are actually providing information about temperature, about, say, your child to your smartphone, and gives you alerts based on, if a child will spikes in temperature. But from that data, we'll also collect huge amounts of information at the population level. We're also thinking about how to integrate other types of data sources into our Zika surveillance efforts. I'll quickly run through this, but the idea is that we're now looking to take data not only from official governments, but we're in our collaboration with Google, taking data on search query data, so what people are searching for online in terms of symptoms, along with what people are reporting on Twitter, to make actual predictions of what we might see going forward in Zika, in terms of Zika. So we're developing forecasting methods that essentially allow us to understand, okay, so in Colombia we have what people are searching for, what they're saying. Can we make predictions about what is actually happening at the population level? And we're doing this in Honduras as well, where we can actually get quite good predictions 2 to 3 weeks ahead, as well as El Salvador. So we have now a system that we're developing that is combining what people are reporting, but also essentially passively what people are searching for online and what they're actually tweeting about, as an example. One of the things that we're also moving towards is trying to build a better network. And so this is much more from a global health perspective. One of the challenges is that we have this network, we have an inability to verify unknown outbreaks that are happening around the population. And so we're building essentially an informal network of experts, of trained public health and epidemiologists to provide basically validation of disease outbreaks. So for the future Zika that takes place, we want to actually have an ability to respond much more quickly than what we did before. And essentially what we're doing is we're almost called like the bat phone for epidemiologists, where we have, and I'll show you, over 1,300 members so far, and it's growing, that are essentially those that can be quickly there to respond about intelligence that we've gathered. So we find an unknown mysterious outbreak that's happening in a population, we can quickly query the people in that particular area to find much more detail. And this can help support efforts, of course, of the CDC and WHO in their ability to collect information about what's happening on the ground. And I'll just close with one last aspect, and I don't know if people have tried this when they were in, it was in DC last year, but we're also thinking about innovations in response and how to distribute vaccines and other types of interventions, especially in an emergency scenario where it's so difficult to get a lot of vaccine and a lot of interventions to populations through some of the more conventional channels. And this is an effort, of course, we worked on with Uber, where we went to them and with the idea that it's so hard to get people to actually get a vaccine, even if a CVS or Walgreens is within a block. What if you bring vaccines to a person, whether it's Uber or Lyft or anybody else? The idea, it was very simple: put nurses in cars and start to distribute vaccines on demand. And we did this actually in DC, both 2 years ago and last year, as well as 40 other cities. And what we found was the idea of just being able to provide on-demand interventions. So perhaps this will happen with Zika as the vaccine is developed, is a way to get people to do an intervention that they wouldn't do otherwise. And it's not because they were philosophically opposed to the vaccine. It's just that they were just not really up for doing it, or they didn't have access to it. So it's actually a good audience to be showing this to, because this was a particular segment of the population that just doesn't get the vaccine because it's not easily accessed. But with the on-demand nature of it, a lot of people ended up doing it, getting a flu shot for the first time in their lives. So we're thinking about not only just the surveillance perspective, but then once we have this Zika vaccine in place, what is going to be the best way to distribute it? And not only vaccines, but you can imagine in many parts of the globe where we need to do massive distributions of bed nets and other types of maybe non-pharmaceutical interventions, this is a great mechanism to get it out there. So I'll leave it there, thank my team, and turn it over to Dr. Song. Thank you.
Thank you. Particularly like that tweet about winning at being a millennial. That very apt. So our next speaker is Dr. Song, who is the director of the stem cell program at the Institute for Cell Engineering and professor of neurology at Johns Hopkins. And just to let everybody know, there'll be ample time right after they speak for questions and answers for both of you. So, Dr. Song.
Okay, very happy to be here, and thanks for the AAAS to invite me to be here. And it turned out the most challenging here is actually find a parking lot. So it took me a long time to walk over. So actually I'm here today is going to tell you a story. I was trained as a developmental biologist in my own lab at Johns Hopkins University. I've been working on stem cells for the past 14 years. So I was not really a Zika researcher until earlier this year. So basically, we've been working on stem cells for a while, as I mentioned, and there's tremendous progress in the last decade in the stem cell technology. So basically nowadays, everybody in the room, if we can get a biopsy from your skin, for example, cells, we can grow them up and then turn them into stem cells. Eventually has capacity to turn into any cell types in your body. So here it just shows basically, if you look at them, is we can take either from the normal people or patient, and then we turn them so-called induced pluripotent stem cells. Now people figured out a way, actually this is the power of basic science research. People figured out how to turn this into different cell types, for example, the heart muscles, the neurons, here it shows the neurons, and all different cell types. Traditionally, this has been done in the 2D cultures, basically they're in the monolayers, and this for good reason, because this way you can really control the conditions, you can get pure population of cells, and then you can use those cells for testing. So now in the field, people have been using this as an approach to study, for example, how specific cell type are developed, how if you have a genetic defect in these cell types, what caused the defect. And also now, pharmaceutical industry now start to use these cells to screen for drugs. You can imagine this is also, because all these cells are coming from the same, from individual people, it has the same genetic material, so you can think about sort of a personalized medicine. But one of the caveats of this approach is that these stem cells have been sitting in 2D cultures or monolayers. So they don't really have the structure normally shown in the body, which is 3D, and they also have cell-cell interactions that form the complex. In the last 2 years or so now, people start to sort of think about, can we actually make an organ out of these stem cells instead of individual cell types? So the initial effort come from is instead of doing these cells in monolayers, and they put them in aggregates. So here you can see you basically clump the cells together in the tissue culture well, and then you can embed them in certain biomaterials, in this case matrigels, which are purified from animals. And then once you have them in the cluster, and then you can basically put in this sort of the spinner, bioreactor, spin them. And this turned to be important because in this case is when you spin them, you can increase the oxygen and nutrient penetrance. Eventually you can make much bigger tissue. So this actually bioreactor was initially developed by NASA about in the 1990s. The idea was to try to grow cells in the space in the zero gravity. So here just shows amazingly now, instead of individual cells now, what you can see is you can start grow tissues here. They form a structure just like a developing brain. And the important thing, of course, here is we can do that with human cells. So for the first time, we actually have access to study human brain tissues without really going to the human. So these provide a lot of opportunities. So we started from this a couple years ago. Is really we thought this is a pretty cool idea, and we want to study this in more detail. The problem at the time so here it just shows exactly what it looks like in the lab. You can see this is the bioreactors. You grow the cells, you spin them, and then here has a magnetic spinner. So you have a machine here. You spin the solution. Eventually, you grow the organoids. The problem is for many labs facing is this actually takes a lot of space. Right? So in one incubator, you can see you can only have a few bioreactors. But probably more sort of challenging for lab is we use a large volume. Right? The size is about a Coke can you have. There's a lot of a medium we use to culture the cells. And normally for culture human stem cells, the medium are very expensive because we have to use different nutrients, factors. So that becomes actually not affordable for many labs to do. As a result, this actually leads to the protocol was not optimized because now people start with so-called intrinsic protocols. You just grow the cells in their spin without giving them anything else other than the basic nutrient. And they call them intrinsic protocols. Actually, the real reason is actually, as a scientist, we really cannot afford to give them anything else because it's too expensive to grow them. So that's the starting point. We saw the first thing we need to do is actually, can we make this cheaper, more user-friendly? And for this, actually, it turned out this question was solved by 3 high school students in the lab. So they actually do internships during the summer. And they came over and they saw, well, in the high schools, they have a lot of 3D printers. They normally just print the Legos or other stuff for their fun. In this case now, what they say is, can we actually just make the bioreactor smaller? So what they did is, you can see here, just like the Lego, they can print the gears, the spinners, and then eventually they can assemble them just a Lego. And now you can put them together on a standard tissue culture plates. In the case of tissue culture plates, of course, the volumes are much, much smaller. So we can reduce the volume to about 100-fold. So this way you save the cost immediately 100-fold. So with this approach in hand, we actually now can go ahead and testing. Can we actually get better conditions and grow these 3D organoids? And just show you, finally, we actually succeed, come up with a protocol where we can build the organoid much more representing the developing human brain. So we have stem cell layers. You can see here the stem cell label, and then we also have neural layers. So that's where we were at the time. Is we thought we now have a nice system in the lab where we can study the basic biology of human brain development. And of course, we have been interested in brain disorders. We thought about how can we actually apply this system to study developing brain disorders. So I was talking to my student, and we said, okay, we need a model to study. We can start with a disease with genetic insult. And then basically my student just pointed to his computer screen at the time. So I remember that's January of this year. And you can see a red flash CNN, right? And that's the Zika virus. So that was the beginning of this year. And of course, you all saw these pictures. And the Zika virus you heard is really spreading in South America. And because the virus normally actually leads to very mild symptom, or even majority of people don't have symptoms, so this has been sort of neglected for a long time. And only recently, so actually here shows, this is a drawing actually done by one of the high school students. So he started with the Zika virus in the 1950s, they spread to Asia, and they eventually come back to Brazil. And the only reason this virus is dangerous is because the potential link to microcephaly. Because if you got a flu, you overcome, well, fine. But if the baby got a microcephaly, in this case, it's not simply a smaller head, right? This is just one indication that they have a problem with the brain. There's a lot of other problems associated with a smaller brain, but also, for example, inflammation associated with the brain. So because of that, actually, at the time, it was not clear whether these two things are linked together. You have viral outbreak or microcephaly. Because of the series of the potential global health problem, WHO declared the global health emergency on February 1st of this year. So the time we thought we actually can help. So normally, as a basic scientist, we work in the lab, work on the basic biology. It takes decades, or maybe tens of decades, to translate our knowledge to sort of public health or research or drug. But in this case, we saw this, we actually can use our technology immediately ask a very simple question, which is, does actually the virus directly attack the developing brain or actually through other mechanism? So that's actually what we did. It's actually a very quick experiment. So the idea is we take human stem cells, we turn them into neural stem cells. So those are the cells that eventually give rise part of the brain. So that's the defect in humans. So ask the question, does the virus like or specifically target the neural stem cell? So here shows the greens actually are the staining for the virus. And the red is a marker we're looking for cell death. And what you can see here is the neural stem cell in 2D cultures where you can, many of them are infected. It turns out the Zika virus has a specific tropism. So basically means they like the neural stem cells, they go there, and once they get in the neural stem cells, we can see here's the huge cluster of cells, the green block there. They make the neural stem cells a viral factory. So they make more virus, and then release from these cells, infect other cells. And that's why this is actually so dangerous. So even you have a very little infection, a brief encounter or exposure to the virus, but once they get in the neural stem cells, they make them in the factory, and they make more. So not only they make more virus in these neural stem cells, and they cause them to die. And this immediately leads to hypothesis of the death of the neural stem cells because they normally give rise to neurons from the brain, and that may be the reason can lead to microcephaly. So this actually eventually was a piece used by CDC as a biological possibility to claim or declare a Zika virus can cause microcephaly. But for us, this was not satisfying because this only shows the cells can target the cells in sort of isolation. What happens in a 3D normally developing fetus in the 3D structures? So here actually we turn to the 3D organoid or the mini brain I showed you earlier on. So here shows a normal a brain organoid or mini brain. We can see here there are stem cell layers. These are neural layers. What you can see here is exposure of the Zika virus only for 24 hours. We expose the virus for 24 hours, we remove them, and then we grow this mini brain for another 20 days. What you can see here now is you have the stem cell layers, but you can notice the green cells here are much smaller, thinner. So those are the neural layers. And the reason is because of the dying of the neural stem cells, but also will affect their proliferation, will lead to much more reduced production of neurons. And that's just exactly like in the fetus, or when they actually get the infection of Zika virus, which leads to a thinner of the cortex, and it's called microcephaly. So basically, in this case now, we're able to mimic what's happening in clinic. In babies, they actually get exposed to the virus. Here we have a system where we can study how this happens, but probably more importantly, we can use this system to study screen drugs and study therapies. So more later on, actually, because all this was done in the dish, there's also relevance of what happens in the physiological conditions. And most recently, there are 4 studies published to show that we also can model this in animals. So in this study I show you here, you basically can inject the virus into the pregnant mice, and then eventually look at the pups. It turned out we actually got the same result. The virus somehow can pass through the placenta, eventually get to the baby pup mouse. And then what you can see here is they also infect the stem cells in the developing mouse brain and leads to thinner of mouse cortex. So again, this is actually all evidence converging to show that indeed the virus can target the neural stem cells and cause a defect, eventually mimicking microcephaly. So here become another sort of trillion models where we can use that, eventually can test drugs and therapies. So here I try to focus on one cell type, which is the source of generation of neurons. So how about other cell types, right? So in the developing brain, well, in addition to stem cells and neurons, there also another cell type called astrocyte. So those are the supporting cells, which actually are very important. They actually release factors to support the neurons and support the development of making synapse. It turned out actually Zika virus also target the astrocyte. So here shows one actually sample. This is a sort of clinical sample. There's one patient actually in DC was infected with a Zika virus. Eventually, the pathology from the baby was done. Here actually we got the tissue from Cherry Hill from the Children's National Medical Center. We're just staining the tissue patient section to see where the virus are. It turned out the astrocytes, the supporting cells, are also infected by the Zika virus in this particular baby. And what's important is this cell is actually supporting the brain growth differentiation, but more importantly, probably, these cell types actually are present throughout the life. And what they're suggesting is that not only you actually infect stem cells, they differentiate, and eventually they're gone in the newborns, but you also have a cell type which can serve as reservoir for a virus throughout life. And then you can imagine it may have a large consequence beyond simply a microcephaly. So here is actually where we are now. So as I said, it's the high school student help us design this miniaturized bioreactors, and we can use this to develop sort of mini brains from human stem cells, and now we can modeling what happened to the Zika virus infection. We also have made this version, a so-called tower version, where you can just make them a little bit high throughput and been using this for drug testing. So the question now is where we should go next. So here I actually show you a few steps. Some of what are doing, some others in the field I've been going for. The first one is actually it's point-of-care diagnostics. So you heard John's beautiful talk. Look at how eventually people can figure it out where the outbreak happens. But one of the major challenges for Zika virus is that actually we don't really have a point-of-care diagnostic kit, right? So the way people diagnose that is based on the presence of the virus. So when people actually are infected virus, within a window of a week, you have viral DNA, RNA, which you can detect it by a molecular method. But afterwards, the virus is gone, and then you have to use other approaches. And those approaches are not currently sensitive or specific, because all the approaches using so far can detect both Zika and dengue virus. And that's a problem because in Puerto Rico, for example, majority of people actually has been infected with dengue before. So in a way, actually, many people actually don't even know whether they got infected. And this becomes particularly important because of the mode of transmission by the Zika virus. In addition to the mosquitoes, you can also be transmitted by sexual transmission. So in a way where the point-of-care diagnosis is very important. We need a very simple way to detect whether the people has been infected before or not. The second, of course, is we need effective, safe vaccines. An animal study just published last week to show that now it's possible to make safe vaccines. And I just heard on news today is actually clinical trials are going to happen very soon in WashU in a few weeks to test the vaccine. But that's going to take time. It's not going to come. So that comes to the next question. If we don't have vaccine, what can we do if it's now this outbreak somewhere in the US? So one of the thought is can we actually screen for drugs? It's of course too late to develop any new drugs. But what people can do is actually take existing drugs. For example, FDA has approved about thousands of drugs. We can test those drugs and test them when any of them actually are effective for antivirus or protecting neurons. So those could be first used, at least temporarily, to sort of for protection. Of course, those also need to go through clinical trials, but because they're approved for other uses, it could be much faster. The third is not only Zika virus has linked to microcephaly, but also has been linked to other neurological diseases, disorders. So it's actually very important now to establish whether that's true. It's a causal or simple association. So that needs a lot of clinical studies, but also need a basic science research to provide possibility. Finally, of course, we need to know a lot about the basic biology of this virus. For now, for many years, this virus has been neglected because we just think it's just like flu. It's not that important. So that's actually where we are. And so this really has been sort of a roller coaster ride for us. As I said, I'm not really a virologist. We're a stem cell biologist. But we're fortunate to be able, really the public, many researchers are able to come together very quickly and to address this global health emergency. So at Hopkins, we actually have many centers, department, investigators have been working together. And in addition, we've been fortunate to have many collaborators actually outside. Specifically, we've been working with Puerto Rico CDC because they have been hit the most, and we've been working with them to develop diagnostic kit using the patient samples. Thank you.
So thank you both very much. So we're going to open up to question and answer, and actually I would like to take the opportunity to ask the first one. You guys have already listed some actions that we can take. Could you summarize any of the low-hanging fruit? You have an audience here of policy professionals in Congress. who are looking for things that they can do to help. What would you consider maybe the first few low-hanging fruit that Congress and federal legislators could do to help tackle both the diagnostic and treatment that you guys have spoke about for Zika?
Well, the first thing is actually, of course, is nobody. I think a very few research institute and labs actually are funded to do the Zika research, right? Because why not? Because this was just a very new thing. So we've been really turning all the other resources to do this, and in a way, we're doing a very limited scale. So of course, if there are sort of this collective effort, we can identify low-hanging fruit and put concrete effort, we can make a lot of progress. And to my mind, I think it's very important to really push through to get a diagnostic point of care, because that's really opened up really how serious this is and how to protect ourselves. The second I thought is can we actually get to the sort of some kind of defense of drug with a already approved, just in case we can use. So those to me are low-hanging fruit.
Yeah, I mean, absolutely. And then even from a basic public health perspective, arming local, state public health with the resources they need around mosquito control programs, but then also surveillance efforts. So we saw this was very effective with West Nile, where there are efforts to collect mosquitoes and actually do testing on those mosquitoes as early sentinels for potential activity of the virus. Those are sort of severely underfunded programs. So this is really, there is of course sexual transmission. This is a mosquito-borne virus. We need surveillance of the vector. And so focused efforts to basically test mosquitoes and collect mosquitoes is that's easy to do. We know how to do it. It just needs to be supported.
Questions? Go ahead.
My question is about Guillain-Barré, so it's neurological. We know that Guillain-Barré is not uncommon after exposure to pathogen, viral or bacterial pathogens. So it's not, you can get Guillain-Barré after dengue and West Nile, etc. The interesting thing is there's been a lot of research done in China that people who were exposed to these pathogens and got Guillain-Barré had a certain HLA haplotype. And since we have a semi-closed population from British Polynesia, I haven't heard of anyone, and maybe you have, looking at the relationship between HLA haplotypes and susceptibility to Guillain-Barré. Because if Zika does get into the U.S., and we could see an increased number of cases of Guillain-Barré.
So that's actually a good question, right? So clearly the causality was not caught by the Zika virus Guillain-Barré. I think the reason is we don't have a larger enough numbers or biological reasons, sort of possibilities. But if you look into the case in Brazil, you have more microcephaly, but in Colombia, you actually have a lot more the other problems. So clearly, they could be really a genetic factor there as well. In China, clearly, they don't locally, I think except Hong Kong or South, they have
There's a huge number of studies done in China.
No, for Zika, I mean.
No,
yeah.
not Zika.
But for others, clearly, so
Exposure from the pathogen.
exactly. So clearly, there are multiple salts can cause the same problem, right? So the question is how they link together. It's the same thing for microcephaly. There's a genetic form of microcephaly, but they're not identical to the Zika-induced microcephaly. It's just the name, but they are not the same thing.
Yeah, so you talked about controlling mosquitoes, and one of the new prospects, obviously, is gene drive.
Yeah.
And so what is your take on this sort of experimental stage with gene drives and,
Yeah.
say, making all male offspring for mosquitoes?
Yeah.
Do you,
I mean, these are all, exciting sort of prospects that they've been exciting for, well over a decade now. The idea that you could drive certain genes in populations to drive infertility, or there's also symbiotic relationships. Well, I don't know if you've heard about Wolbachia as a bacteria symbiont that can potentially deactivate or prevent transmission of virus. So yeah, many different sort of promising areas. Of course, huge amount of ecological concern about when you drive these things in populations, what are the unintended consequences? I think there's been some very early studies that have shown some level of success, but the larger scale implications are not quite there. So yeah, I mean, these are not sort of short-term necessarily opportunities, to deal at the continental scale. But I think, of course, from a funding perspective, And as we think about, mosquito-borne viruses, and Zika is not going to, there's a long list of viruses. If you read the literature from the same time period where Zika was discovered, there are many, many other viruses that look similar that we have yet to see emerge. So just thinking about these broad-scale, potentially new novel control methods, of course, are going to be important, whether it's for Zika or whatever's coming next, because we will be talking about another vector-borne disease in the next couple of years. I can guarantee you that.
Dr. Brownstein, you talked a little bit about the Olympics. Do you have any models and forecasts for how it's going to spread post the Olympics?
Right. So I think it's a good question. We're still building those now based on historical Olympics travel and also using the World Cup as an analogy. I think what's good about the timing of the Olympics is that it's not going to be the height of mosquito populations. So right now the thinking, at least, is that we're going to have a small number of cases. I mean, these are just based on the existing models and what we know about the sort of rise and fall of mosquito populations in the area where the Olympics are taking place. So that is reducing our sort of major concern for global spread of Zika. But so we're still building those transportation network models. So we don't have necessarily a priority list of sort of cities that we might expect to enhance surveillance on just yet.
How do you factor sexual transmission into your model, or do you?
Well, I mean, so from just a pure transportation network side, what we're thinking about is just sort of the idea of the importation of a case, regardless of whether that then from there becomes an individual that's bitten by a mosquito or involved in sexual transmission of the disease. So that, from that perspective, that risk assessment's about the same, but it definitely is different in terms of where we think spread might take place, because a lot of our models are based on then subsequent chains of transmission based on environmental suitability, and that doesn't take with that mode of transmission, that's not a factor. So we definitely have perspective where we might see export, but beyond that, we're going to have to think about other types of models. I think there's still the jury's out about how likely that is too. I think we have seen these cases take place, but I think that we still need a lot of data, and I think that's another area of funding, is just to understand the likelihood of transmission, because I don't think we have a good grasp of that just yet.
Just this week, the Brazilian strain was identified on the west coast of Africa. You have that, so you're right. Can you tell us what your forecasts tell us about the spread through Africa?
Yeah. I think that's another area of research that's really challenging, because as far as we know, there's actually been a lot of transmission on the African continent for many years, but we just have never had the surveillance capability, right? So originally this virus was discovered in Uganda. Likely it's been around, but nobody's been monitoring for it. And because symptoms are so mild, although of course the implications to babies is pretty severe, I think there's an unknown sort of burden that's existed. And then from there, the idea is, well, how immune is the population already? So we don't have baseline understanding of the immunity that exists, because of course with an immune population, like we've seen for other viruses like West Nile, we'll see these sort of epidemics wane over time just because there's just not a susceptible population that exists. But once again, we don't have that baseline data, so it's just very hard to predict. Not to mention that a much more important virus is, I wouldn't say more important, but equally important, a yellow fever is spreading pretty rapidly, where there is actually a vaccine to control it. So just make a plug for the fact that we should also be turning our attention to what's happening in parts of Africa like Angola around yellow fever, where there's an opportunity to control a virus that's having devastating consequences.
I just had a question about data mining for queer researchers. So,
Yeah.
like when people say like I'm feeling these kinds of symptoms or something, how does your team kind of distinguish between those who are like hypochondriacs or like those who are
Right. No, it's a good point, and something. So we work closely with Google for a lot of years on, and there's a lot of concern about the Google Flu Trends. I don't know if you remember that tool. It sort of was sunsetted, although we've sort of taken the reins a little bit. It's challenging for sure, and being able to find the right set of terms that relate to someone actually querying about a set of symptoms rather than sort of just informational. And there are ways to separate these types of terms, and very specific terms people will use when they have symptoms. Now, of course, there's going to be a challenge when it's very broad set of symptoms. that people are just going to be querying them and they're not necessarily related to the disease of interest. And then from there, we have to have baseline level models that essentially are able to distinguish sort of signal from noise. And so you're going to have a lot of queries no matter what on those terms, and you're just having to look for sort of signals above a certain amount of threshold.
Patterns that have like correlated with.
Absolutely, yeah. So we use sort of historical official data to help build the models.
Any further questions?
So when you look at the fetal brain development stuff that you've looked at, what theories do you have at the moment about why the prevalence of microcephaly, while way higher than background, is still not that high? Is it because of severity of infection, viral load, and what something about how it passes through the placenta, and the babies that have it pass through the placenta almost always have microcephaly, or just we don't know yet?
So I think that we don't know yet. So one of the sort of guesses is passage through placenta, because in the case of that's actually in the case of animal study has been very useful. So in the case of animal studies, if you just inject the virus in the normal mice, most of them actually don't have any problem in the pregnant mice. But if you use immune deficient, use some where they don't have the mechanism against the virus, then you have much larger cases of problem. So in humans, it could be this particular population are more sort of at risk. But we think mostly if the virus gets into the brain, it's actually very infectious. The virus is very infectious. So the question is, could they pass or not? The exposure time is also could be a issue as well. Is how long, how healthy, how strong the mom at the time, we think. But mostly we think is a passage through the placenta is the limiting factor.
And is there I assume there's research, but maybe not, there's not enough funding into what these vulnerable populations and more vulnerable would be?
So there's no, actually back to your question, there's no genetics has been done before. There's not really enough study have done that. People start looking into, in the animals, is sort of look at how the cells actually get into through the placenta, what are the cells that are infected, how can you block that, right? So that's the other issues. But it's very early, right? Think about it, is majority of the field come together this year. So it's only 6 months doing this. It's already amazingly fast in terms of science research.
Is there concern with accuracy in gathering data from populations that don't have internet access?
Oh, yeah, so, sorry, I thought we were bringing it. Yeah, absolutely. So that's something we've always struggled with because our data is as good as the access to information technology is. So of course, there's biases. So we try to avoid making sort of population level estimates because of the fact that there's major diversity in levels of access. And for sure, we're going to have access to certain parts of the globe with data much better than other parts. So for instance, not only access, but also just in terms of information freedom. So the Middle East, for instance, is a big, blind spot for us in terms of the kinds of information we collect, especially as we focus on MERS surveillance. That has been a real problem because we don't have a lot of data coming from populations from social media. So for sure, we have to correct for the fact that we don't have uniform coverage. Question?
Well, if you please join me in thanking our speakers.